Welcome everyone to the Cancer Models and Mechanisms meeting. My name is Elaine Martis. I'm usually in Columbus, Ohio at Nationwide Children's Hospital and the Ohio State University, and I'm one of the meeting organizers this year. So it's really wonderful to be able to welcome everyone here in August, which in my opinion, since I come here about 4 times a year at different times of the year, is the best season of the year to be here. And I think the weather is not going to be a disappointment, although we are going to be huddled in here in the dark looking at slides some proportion of the time. It is what it is. So just a few quick facts for those of you who like facts, and we basically all do. At the meeting this year, we have close to 420 registrants, 419 to be exact. 384 people are in person and 55 are virtual. Too bad for them. Much better to be in person, but I'm sure they'll enjoy listening from afar. There are going to be 70 talks, so get ready, and 178 posters. The posters, if you haven't been here for a while, happen in a new pavilion that was built a few years ago. You just walk down the path here in front of the auditorium, and it just appears in front of you. It's a beautiful place. Called Beyond Hall. And so that's where we'll have our poster sessions. Couple of quick things. You got an email welcoming you to the meeting. There's a Slack sign up there. If you haven't signed up for the Slack channel, please do. First of all, as an older individual, talking on Slack makes me feel really hip. If you don't fall into that category, it's also a great place to interact with other people at the meeting, and for virtual attendees, that's how they'll be asking questions, so you'll understand what questions are being asked from the virtual attendees. You'll also get any updates in terms of last-minute cancellations or changes, etc., which we can't capture in a static book of paper. Although I got to say, the front cover this year is lights out. You'll learn more about that later. It's from some work that we're just getting published soon, so I'm completely biased, full disclosure. There is going to be something to look forward to after tonight's talks. If they alone aren't enough, there's going to be a happy hour here in Grace Auditorium, sponsored by Revolution Medicine, immediately following the session. So leave the session, go outside, grab a cocktail, get to know somebody new, or catch up with somebody you already know. There's the traditional wine and cheese, which we all look forward to. That's going to be tomorrow night, Wednesday at 5. It is also going to be on the lawn outside Nichols Beyond Hall, which is where the posters are being presented. Beautiful, huge lawn. Set up there is gorgeous, and I think the weather is not going to disappoint. So yet again, a great opportunity to get to know new people or catch up with people you haven't seen in a while. If for some reason the weather doesn't work out, we will have that in the auditorium and patio out here that's covered by the marquee just outside of Grace Auditorium. And there will be another announcement if anything changes, so don't worry about that. Finally, and perhaps most importantly, after the lights out dinner to celebrate the meeting and its success on Friday evening, there will be a DJ and dance party in Bush Lecture Hall, which is right next to Blackford Hall, which is where the cafeteria is located. If you've been coming here for a long time, like some of us, that's where posters used to be presented. So there'll be a DJ and an opportunity to dance after you've had way too much wine to drink at the banquet. Something to look forward to, scientists dancing, always entertaining. Please keep your video cameras to yourselves. We don't need that. So that's all for my announcements. I'm going to hand off to my good friend Alejandro Sweet Cordero, who's co-chairing this session, and we'll get things started with the magnificent Ben Neel. Thanks, everyone.
All right, so we're going to get right to it and get started. And our keynote speaker tonight needs no introduction. Benjamin Neel, who's going to talk about his old friends doing new tricks.
Okay, before we get started, I've been informed that it is the birthday of one of the organizers, okay? And so we have two choices. We can either sing Martin Happy Birthday right now, or we can sing, for those of you who are old enough, maybe Elaine can help, When I'm 64 by the Beatles. So I think probably it's easier to sing Happy Birthday, so I think we should sing Happy Birthday to Martin, and this is not coming out of my time. Okay. Okay, so happy birthday to you, happy birthday to you, happy birthday, dear Martin, happy birthday to you. Okay, so...
say that I am actually no longer an organizer, and my former role as an organizer has been taken over by Monty Winslow and Karen Chakowski.
Well, he always was an organizer, so he could get happy birthday sung to him by more people than he ever has sung to him anywhere else. And also, I did this on purpose because I know I'll get at least one laugh and one set of applause in this talk. So, okay, so I want to thank the organizers for inviting me to give my valedictory keynote. And what I'm going to talk about is a story that's soon to be published on hypoxia-induced inflammatory cell death pathway triggered by PTP1B inhibition. And the reason for my talk is that it's a new surprise by an old friend, the old friend being PTP1B. These are my disclosures. None of them are relevant to this talk, so I won't spend any time on them. So I think in the last 25 years, we've learned some key principles about cancer biology that have implications for cancer therapeutics. The first is that targeted therapies debulk, but they don't cure, ever. Immune therapies cure some, but not most patients, and I think that that's telling us that the only way to cure cancer is to generate a durable immune response to the tumor. But that's a hypothesis and not proven. And combination therapies are almost certainly going to be needed in most cases. So that's led to several challenges. And the first one is, when thinking about these combinations, how do we make cold tumors or cold parts of tumors hot? And how do targeted and other therapies, therefore, affect the TME? And as I said, I'm going to talk about one story today about PTP1B and an inflammatory cell death pathway, but I also want to call to your attention a poster that will be given in the second poster session by Oscar Pundell, who's taking a very systematic way to generate fallopian tube epithelial-derived cell lines that have mutations that are found in human ovarian cancer that are syngeneic and can be used to study interactions between the genome and the microenvironment. I think there's some interesting findings that people who are interested in genome evolution would like to see at that poster. Okay, so the old friend that I'm talking about is actually the oldest friend in the tyrosine phosphatase field, PTP1B. This is a cartoon-like structure of PTP1B. It's localized to the ER via this ER targeting sequence. And the reason it attracted a lot of attention in the 90s was because knockout mice are resistant to high-fat diet-induced obesity and diabetes. Its substrates include the insulin receptor and IRS1, which explains its effect on insulin signaling, but it also has a number of other RTKs as substrates, and I want to call to your attention very nice work that's been done by Nick Tonks here on track B and its possible role of PTP1B inhibitors in treating forms of autism. And the reason why these mice are resistant to high-fat diet-induced obesity, at least in part, is because of leptin receptor-associated JAK2 signaling, which is negatively regulated by PTP1B. But also, more recently, Tony Taganis in particular, and also Michel Tremblay, has pointed to the role of PTP1B in regulating immune cytokine receptors and via JAK2 and TYK2, and that's led to its potential role as a dual inhibitor of PTP1B and T-cell phosphatase, or PTPN2, for immunostimulation. But several years ago, we were very surprised, and this is basically almost a 20-year story. Okay, we were surprised, and the same results were obtained by Michel Tremblay, to find that PTP1B knockout mice were resistant to HER2-mediated breast cancer. And the differences between these two lines, these two results in terms of quantitative differences, have to do with differences in mouse strain and the particular mutation. But in general, the qualitative results are the same. And we never really had a good explanation for why this was the case. And so about 10 years later almost, when Robert Bond was a graduate student in my lab, he found that PTP1B regulated survival during hypoxia, particularly severe hypoxia, less than 0.5%, which occurs in many tumors. And it did this by regulating a protein called RNF213. So what's RNF213? So I know that one of the great things about Cold Spring Harbor, and I already forgot to point out that my first talk was at Cold Spring Harbor about 44 years ago, and so I really like being at Cold Spring Harbor because it's where graduate students and postdocs get to present their work after you hear a few things from old folks like me. What RNF213 is for the graduate students and the postdocs is something that you run like hell when a PI asks you to work on it. Because it's a very large protein. It's about 600 kilodaltons, which means all the standard types of gene transduction things that you want to do with more normally sized genes can't be done. You have to introduce all the constructs by piggyback recombination. It's hell to make mutations in them, et cetera. It's a ubiquitin ligase that has two E3 domains, a ring E3 domain and a so-called RZ domain, and two functional ATP domains. And if being a 600 KD protein isn't enough, it forms a hexamer of about 3.6 megadaltons. Now, SNPs and RNF213 are associated with a rare syndrome called Moya-Moya disease, which causes precocious carotid artery strokes in young adults. And most of these affect the ring domain, and keep that in mind. And especially all the high-penetrant SNPs affect the ring domain. Now, Moya-Moya disease is also associated with autoimmune and inflammatory disorders, so it'll all be relevant for this talk. RNF213 localizes on and stabilizes lipid droplets, and it promotes palmitic acid-induced lipotoxicity, at least in K562 cells. And it's also proposed to ubiquitinate via this RZ domain lipid A of salmonella, possibly on the lipid component and not the carbohydrate or protein component. And it's possible that that's why these SNPs are conserved in evolution, because they probably confer resistance to salmonella. And it acts as an ISG15 sensor. So we had a number of questions about the relationship between PTP1B and RNF213. We had noted, or Robert had noted, that RNF213 was tyrosine phosphorylated and trapped by PTP1B, suggesting it might be a PTP1B substrate. So we wanted to know, how does PTP1B regulate RNF213? We wanted to know how RNF213 regulates hypoxic cell death. In particular, what's the mechanism of cell death in PTP1B deficient and inhibited cells? And is this pathway relevant in vivo? And for us, in vivo means in mice. And I'm going to answer, I think, all of these questions today. And every experiment that I'm going to show you, except for the actual mass spectrometry, was done by a very hardworking postdoc, Abhishek Bhardwaj, who's now on the job market after his paper has been accepted. And you can see here how hardworking he is because he looks exhausted. Okay, so first, how does PTP1B regulate 293? I'm already 3 jokes in, and they're all working, so this is good. Yeah, okay. So we did mass spectrometry on tyrosine phosphorylated RNF213 and identified a phosphorylation site, tyrosine 1275. And we think this is the critical phosphorylation site that's regulated by PTP1B for reasons that I'll show you in the next slide. But if you look on the cryo-EM structure of monomeric RNF213, tyrosine 1275 is located on this hinge between the N-arm and one of the triple ATPase domains. And so it's in a position that you might, first of all, think would be accessible to tyrosine kinases, which is always good, and also might be involved in regulating the enzyme. Okay, so why do we think this is the critical phosphorylation site for interacting with PTP1B? Well, if we take BT474 HER2-positive breast cancer cells, knock out RNF213, and reconstitute and also express in them an HA-tagged version of a substrate-trapping mutant, PTPN1D-A, then you do an immunoprecipitation, then that brings down RNF213. So wild-type RNF213 is trapped by PTP1B substrate trapping mutants. But this single mutation in RNF213, 1275F, you lose that association in either direction in an immunoprecipitation reaction. And I won't go through all of the data, but we believe that this phosphorylation controls oligomerization, or at least dimerization of RNF213, because if we express a flag tag version of wild-type RNF213 and a GFP version of wild-type RNF213, we can bring down each of the proteins by precipitating one and blotting for the other. But if we mutate that single residue, 1275, we substantially impair the co-immunoprecipitation. In terms of the kinase, I'm not going to go through all the data for this, but we screened for kinase inhibitors that might block hypoxia hypersensitivity, and we found that ABL inhibitors, including the highly specific allosteric inhibitor asciminib, which only inhibits ABL1 and ABL2, basically block hypoxia hypersensitivity caused by PTP1B. I won't show you the data, but we also showed that knockdown of ABL1 and ABL2 blocks hypoxia hypersensitivity, and that ABL1 phosphorylates RNF213 in vitro, and that phosphorylation is blocked by mutation of tyrosine 1275. So this is a complicated story, so I'm going to give you a summary all the way through to make sure everybody's on the same page. So what I've told you so far is that RNF213 is reciprocally regulated by the kinase ABL1 and possibly ABL2 and the tyrosine phosphatase PTP1B to control phosphorylation of tyrosine 1275, and the tyrosine 1275 phosphorylation controls oligomerization of RNF213, possibly into an hexamer, but definitely at least into a dimer. So that leads to the next question that I'm going to address, which is how does RNF213 regulate hypoxic cell death? Okay, so here we first thought that, well, let's find out what RNF213 interacts with. So Abby made two different forms of turbo ID-mediated fusion proteins, and the one that works best for identifying interactors is the C-terminal fusion, RNF213 turbo. And reassuringly, we found that this fusion protein, when you add biotin, specifically biotinolates PTP1B, very gratifying, and also USP15, which had been previously reported to be a stable interactor of RNF213, and we also have the same data in our lab. But interestingly, we immediately called our attention was immediately called to two other proteins, CYLD and SPOT-A2, which are major deubiquitolase complex for NF-kappa B. And we thought that this was really really important, because if we knock out either SPOT-A2 or CYLD alone, that this confers dramatic hypoxia hypersensitivity in the presence or absence of PTP1B knockdown. Okay, so what I've shown you is that RNF213 is regulated reciprocally by ABL1 and PTP1B. That leads to hexamerization. And that raises the obvious question, since RNF213 interacts with SPOT-A2 and CYLD, does RNF213 ubiquitinate CYLD and SPOT-A2? And if so, which domains in RNF213 are required? Okay, so Abby set up an in vitro ubiquitination reaction using UBE1 as the E1 and UBE2EL3, which we and others had shown was a good E2 for RNF213. And you can see that in this in vitro ubiquitination reaction, RNF213 directly ubiquitinates recombinant CYLD and directly ubiquitinates recombinant SPOT-A2. And then we wanted to know what domain. Remember, I told you there's two potential E3 ligase domains, the classic ring-like E3 ligase and the RZ domain. And we were surprised to find that if you mutate the RZ domain, which is supposed to ubiquitinate lipids or carbohydrates in lipid A, you lose ubiquitination of CYLD, and you also lose ubiquitination of SPOT-A2. What happens when you use a ring domain? So when we made the ring domain mutation, we were really surprised, because actually when you mutate the ring domain, you get increased ubiquitination of CYLD and increased ubiquitination of SPOT-A2. And you'll recall that I told you that the high penetrant SNPs of Moya-Moya disease and autosomal dominant syndrome, okay, are almost always in the ring domain. Okay, so this data suggested that the ring domain might negatively regulate the RZ domain and give you a gain-of-function mutant for the RZ domain. And so Abby made a double mutant, the double mutant in the ring domain and the RZ domain. You can see you lose the ubiquitination of SPOT-A2 and CYLD. So is any of this relevant in vivo? That was, or at least in cells, that was all in vitro in a test tube. Okay, so again, Abby made RNF213 knockout cells. He's done this in other cell lines as well. I'm just going to show you for one, but the paper and the preprint that's online has all of this data in it. If you can see that in RNF213 knockout cells, you accumulate CYLD and SPOT-A2. If you restore expression of RNF213, you substantially reduce the levels of CYLD and SPOT-A2. If you restore expression of the ring mutant, which again I told you is a gain of function for the RT domain, you basically eliminate CYLD and SPOT-A2, and any of the double mutants, the single mutant in the RZ domain or the double mutant in the ring in the RZ domain, lose the ubiquitination and therefore lose the degradation of SPOT-A2. And this correlates directly with sensitivity to hypoxia. I'm not going to go through all of these mutants here, but you can see that if you have RNF213 intact, you get hypoxia hypersensitivity. If you knock out RNF213 and just have the vector there, you lose the hypoxia hypersensitivity. If you restore wild type RNF213, you restore hypoxia hypersensitivity, but you can see that there's actually more hypoxia hypersensitivity, for example, if you use these ring domain mutants, and it goes away if you make the RZ domain mutant. Okay, so to summarize what I've shown you so far, RNF213 is reciprocally regulated by ABL1 and ABL2, leads to oligomerization and activation of the RZ domain, which is inhibited by the ring domain. That leads to SpotA2 and CYLD ubiquitination. And I won't show you these data, but you also need the Lubac complex, as you do in Salmonella, and that goes to gives you endolysosomal degradation of SpotA2 and CYLD. It's all in the paper. That leads to the next question, which is how do hypoxic PTP1B deficient or inhibited cells die? So we were surprised when we tested a panel of inhibitors of cell death that general caspase inhibitors like ZVAD, FMK completely blocked hypoxia hypersensitivity. And that was surprising to us because Robert Bond, who's very careful, when he was in the lab, had found that there was no increase in apoptosis in RNF213 in PTP1B deficient cells that was RNF213 dependent in hypoxia. But Abby actually realized that general caspase inhibitors like ZVAD also inhibit inflammatory caspases. And remarkably, use of the inflammatory caspase inhibitor VX765 also completely blocked cell death in hypoxia in response to PTP1B deficiency or inhibition. So these stats suggest that PTP1B cell death in hypoxia was an inflammatory cell death. Okay, so that immediately suggested to us, since we knew it wasn't necroptotic because we also checked necroptotic inhibitors, and we knew it wasn't ferroptotic because we checked ferroptotic inhibitors, that this was pyroptosis. So this is a simplified cartoon of all the different ways that you can get pyroptosis in cells, but that's too complicated for me. I can't even use SLAC. I'm older than Elaine. So this is a simplified version for a PI. And basically, the critical thing in pyroptosis is that there's two steps. One, a priming step, in which the inflammasome is assembled using inflammasome compartments. And then a second activation step, where the inflammasome is activated. That causes caspase-1 cleavage and also cleavage of pro-IL-1 beta to IL-1 beta and release, and also cleavage of gasdermins, which insert into the membrane in a palmitate-dependent fashion and cause a pore that leads to pyroptotic cell death. So first, let me convince you that these cells die by pyroptosis. So if you knock down gasdermin D, which is the major gasdermin that's relevant here in these cells, you completely abrogate hypoxia hypersensitivity caused by PTP1B deficiency. So this is a pyroptotic gasdermin D-dependent cell death. And again, if you look at IL-1 beta release, this confirms that it's also pyroptotic cell death. Again, if you look at BT474 cells, you get increased IL-1 beta production. If you knock out RNF213, you lose it. You restore wild type, you get it back. You put the ring domain in, you're leaking IL-1 beta even when you're not hypoxic. And all of the other mutants lose it, as you would predict based on this model. Okay, so RNF213 oligomerizes in a PTP1B ABL-dependent fashion. The ring domain inhibits the RZ domain. When the RZ domain is activated, it ubiquitinates and degrades SpotA2 and CYLB, and through the Lubac complex, you get degradation of these, and that leads to an obvious suggestion about what the priming step is, because I told you at the beginning that SpotA2 and CYLD are a major deubiquitinase complex that's a negative regulator for NF-kappa B. So that suggested that the priming step was NF-kappa B activation. So is it? So again, if you transfect in an NF-kappa B reporter, you can see that in BT474 cells in hypoxia, NF-kappa B activity is increased. In knockout cells, you lose NF-kappa B activation by hypoxia. You restore it with wild type. Look at what happens with the ring domain mutant. You have a lot of NF-kappa B activity, even basally, and all of the RZ and ATPase domain mutants lose it. Okay, so what's the second signal? So here, again, we realize that hypoxia is not just about HIF. So everybody knows that HIF is the major hypoxia regulatory pathway that directs hypoxia-dependent gene expression, won the Nobel Prize and everything like that. But in addition, at more severe forms of hypoxia, and recall, I said this only works below 1% oxygen, you activate other hypoxia response pathways, particularly ER stress pathway. And the ER stress pathway is a known inflammasome activator pathway. So this suggested immediately to us that hypoxia was providing the activation step by activating ER stress. And we have much evidence that ER stress is activated in our conditions. I won't show you that, but let me show you that it's important. So if we treat with either an IRE1 inhibitor or a PERK inhibitor, we completely abrogate hypoxia, hypersensitivity, and PTP1B deficient cells, arguing for both of these ER stress pathways being important for driving inflammasome activation. So that says that ER stress is necessary for hypoxia-induced cell death in this system. Is it sufficient? So again, if we knock down CYLD and SPOT-A2 to prime the inflammasome, and now we just deliver an ER stress signal with either tunicamycin or thapsigargan, we get cell death in normoxia. Okay, so does any of this matter in an actual animal? Okay, so again, Abby took PTP1, made a variety of different cell lines in HER2-positive breast cancer cells, 2 different HER2-positive breast cells. I'm showing you the BT474 results. And you can see that as we have reported previously, if you knock out PTP1B, you get much smaller tumors than if you have wild-type PTP in the PTP1B. And if you look at those tumors, they are highly necrotic, despite the fact that they're very small. So that's exactly the opposite. Usually you get a necrotic core when you have a large tumor, but these very small tumors are heavily necrotic. But if you knock down CYLD, remember that causes hypoxic cell death by itself, you also get necrosis, and if you knock down both, you get necrosis. But the really killer experiment is this one. If you knock out PTP1B and you also knock out NLRP3, which is the inflammasome component that's activated by NF-kappa B, that is completely epistatic to the PTP1B knockout, and you get a big tumor back. So that shows by genetic means that this hypoxic pathway, this pyroptotic pathway, is critical for the tumorigenesis that we saw earlier. And this correlates with activation of Gasdermin D in the settings where you would expect Gasdermin D to be activated, and caspase-1. So finally, do any of this mutagenesis that we did in vitro, does that translate to in vivo? So again, Abhishek reconstituted the RNF213 knockout cells with all of the mutants that I told you about on the earlier slides, and I'll just point to some critical ones. First of all, if you knock out RNF213 by itself, okay, and you don't restore it, you basically lose the you lose the effect of hypoxia hypersensitivity, and the tumors grow big. If you restore RNF213, that's the red line, you can see that you get smaller tumors again. But look what happens when you restore the ring mutant. If you restore the ring mutant, that's hyperactivated. You get even smaller tumors. And if you make the RZ mutant, you lose or the ATPase mutant, you lose the effect of hypoxia hypersensitivity. And again, all of these correlate exactly with Gasdermin D cleavage in the tumor and cleave caspase-1. So, very complicated story, but here's the summary in a pictorial form. PTP1B and ABL1 and ABL2 reciprocally control tyrosine phosphorylation of 1275. When tyrosine 1275 is phosphorylated, you get oligomerization, probably hexamerization of RNF213. That leads to activation of the RZ domain, which is normally repressed by the ring domain. Activation of the RZ domain gives you ubiquitination of CYLD and SPOT-A2, which in combination with the Lubac complex leads to their degradation, activation of NF-kappa B, and the production of inflammasome priming, and then a second signal delivered by hypoxia-induced ER stress leads to activation of the inflammasome and pyroptosis. So for those of you who are like me, left brain dominant, here's the written summary. So tyrosine phosphorylation promotes RNF213 oligomerization and is reciprocally regulated by PTP1B and ABL1 and 2. RNF213 ubiquitinates CYLD and SPOT-A2 and promotes their degradation. CYLD and SPOT-A2 knockout cause hypoxia hypersensitivity, and the RZ domain of RNF213 promotes pyroptotic cell death under these conditions. NF-kappa B signaling provides the priming signal, and that's activated via RNF213, and then a hypoxia-driven ER stress provides the activating signal for pyroptosis. And the ring domain inhibits the RZ domain, and that has really interesting, I think, implications for moya-moya disease pathogenesis, which I told you is the big SNPs are in the ring domain. And finally, why does any of this matter? Well, because I told you at the very beginning that the critical thing is we need new ways to turn cold tumors hot. And since these data suggest that either PTP1B or CYLD SPOT-A2 inhibition might do that, and that means that in addition to the cell non-autonomous effects of PTP1B and T cell phosphatase inhibition that is being tested in the clinic right now by AbbVie, that there may be important cell autonomous effects of PTP1B inhibition, and maybe PTP1B inhibition alone might have some benefits if the combination of T cell and PTP1B inhibition is too toxic. So I'm going to stop there and again say that every experiment here except the actual mass spectrum was done by Abhishek Bhardwaj, who's on the job market. We were assisted by a number of cores in providing some of the figures that I showed you, and this is our support. And the paper is on BioRx, an earlier version of it, but it was just accepted in Nature, so hopefully it will come out sometime in the next several weeks. So thanks again, and I'll be happy to take any questions. Whoa. I'm going to get some of my own medicine, I see. I didn't have a third cup of coffee. Was that a plant? That was a plant.
Here's the question.
A real question.
Thanks for the talk.
Where is the question?
It's here.
Oh, hi.
It's very interesting that the activations of inflammasome, you know what, it will also activate the immune system. So all of this data showing in the xenograft models. So we will be curious to know what's your opinion in the syngeneic mouse.
Are you reviewer 4? We're doing those, obviously doing those experiments. Reviewer 4 thought the results were too preliminary, that we should do that too. We're doing that. I think you're absolutely right. It's really critical. This is everything I put on the bottom about turning cold tumors hot. That's a hypothesis. But yeah, we are doing that in syngeneic models. Karen has a question.
Is this specific to HER2?
No. Yeah, so our original paper, we thought it was specific to HER2, but it is definitely not just HER2. It's several triple-negative breast cancer lines do this. Head and neck cancer cell line does it. Some, but not all, pancreas cancer lines do it. So we don't know what the determinant is. One possibility is that it's the expression of Gaz-Dermin D. The other possibility is it has to do with whether you can activate NF-kappa B by inhibiting COLD and SPOT-A2. So we don't know what the molecular determinants are, but it's not just restricted to HER2.
Ben, does this have any implications for people that are taking imatinib for CML? Because obviously there are patients that have been on imatinib or similar types of ABL inhibitors for many years now. I'm just wondering if there's any sort of epidemiologic data that suggests that this might be this
that's a
pathway
really
is
good
being
question.
regulated.
I have to say I didn't think about that. I should talk to Brian. Yeah, so you would expect that you would block. Yeah, so that would, if anything, make you less likely to activate the inflammasome. So that's a really good question, though. Yeah, where am I looking?
Right here. Great talk.
Hey, Dave.
Ben, that was great. What happens to all the pro-survival pathways that are activated by hypoxia? Is this more like anoxia, and so you don't get the pro-survival?
It's about, we find that depending on the cell line, it's below 1%. To 0.1%. So, I don't know how, I think if you activate Gaz-Dermin D, that will kill a cell no matter how much anti-apoptotic stuff you have there. But I must admit, there's probably people here who have forgotten more about cell death than I know. So maybe they can correct me. So I think if you activate pyroptosis, if you activate Gaz-Dermin pores, the cells die, regardless of what the hypoxia pathways are, I think. But I could be wrong. I like to be proven wrong, so if I'm wrong, just tell me.
Is the RNF mutant disease, the moyamoya disease, is that driven by pyroptosis? Does that manifest there?
So I don't know, but I will tell you that there was a paper that we heard about because the guy came and gave a talk when I was out of town, but Abhishek went, and then appeared in Nature later, which shows that actually cerebral vasculature, like the carotid circulation in the brain, has the highest levels of Gaz-Dermin D that he's seen. So I think it's interesting to speculate that actually that is important for the mechanism of, atherosclerotic death. And atherosclerotic death, I'm not an atherosclerosis expert either, but I understand that is also inflammatory cell death in atherosclerotic plaques often. So I do think it's likely to be related, and obviously one of the things we'd like to do is follow up on the implications of this study for other types of diseases like inflammatory diseases. You have one more question here, and then maybe we should. I'll repeat it. So.
Any therapy-resistant mechanism happen in any of your pathway?
Yeah. So the question is whether we see any evidence of therapy resistance. Well, in answer to, as I answered in response to Karen's question, we know that not all cancer cell lines that we tested are sensitive to hypoxic cell death in response to PTP1B inhibition or knockdown. So, there obviously are resistance mechanisms. What they are, we don't know. So we haven't carried these mice. We haven't attempted to find such mechanisms. I think I should say that Abby's looking for a job, and my feeling is this is his. He did all this work and suffered intensely. I have a former postdoc in this room who knows that and can attest to that. So yeah, this was a 7-year project, and he stuck it out. So he can do whatever he wants with it. Okay, thank you.